So good afternoon, everyone. My name is Lotfi Merabet. I am an associate professor of associate professor of ophthalmology in the Department of Ophthalmology in Mass Eye and Ear and Harvard Medical School. I have the pleasure today of talking about a project in collaboration with the Leukodystrophy Center at MGH in Boston. And to start off the presentation today, I was asked to speak specifically about the condition of cerebral visual impairment and the development of assessing functional assessments for this condition. I think the most important thing starting off is to state that this is a project that we didn't look for. It was actually a project that found us. And I think that has considerable public health significance and an evolving concern. The most important thing to think about in terms of my presentation today is to think that the #1 individual cause of pediatric blindness and visual impairment is not due to disease of the eye, but it's rather because of developmental damage to the brain. Now, understand that this makes a fundamental shift in our thinking in terms of how we assess, how we diagnose, and educate children who are born with visual impairment. So again, a fundamental shift in thinking. So much so that the National Eye Institute a couple of weeks ago has released their recent strategic plan, and CVI, cerebral visual impairment, has really become front and center in terms of a priority. You can see a recent presentation here by Dr. Michael Chang, the new director of the NEI. A great paper or piece that came out from the NEI recently as well, as you can see from this title, Vision Loss in Children Whose Eyesight May Be 20/20 Requires New Diagnostic and Teaching Strategies. And you may ask, well, how can you use the expressions vision loss in 20/20 in the same phrase or the same sentence? And the answer is that there's much more to visual impairment than just visual acuities. I hope to convince you. Before I start off, I always like to talk about an analogy about the visual system, try to understand how diseases or impairments at the level of the eye are fundamentally different to diseases or impairments at the level of the brain. And the best way I think I can explain this to you is to think of your visual system as a digital camera, which I'm sure everyone has had the chance to use. You'll notice that a digital camera has two really fundamental components to it. There is the lens, which is the optical component, which controls the focusing of an image, right? The clarity of the image, as well as how much light comes into the camera. And the second component is the camera body. And this is the processing part. This is where where the images come in, or light enters the camera, the scene or the visual scene is analyzed, and after you squeeze the trigger or the shutter, there should be, if everything is working correctly, an image that corresponds very closely to what you were looking at, right? The human visual system is very, very much the same. The eyes represent the optical component of the system, right? Responsible again for focusing and letting light into the brain. And the second piece, the processing part, is the brain itself. And this is, in my mind, where the magic happens, right? This is when light enters the brain. This is where the analysis actually happens and allows you to perceive the visual world around you. The reason why it's important to make this distinction is because problems at the level of the eye are not the same as necessarily problems at the level of the brain. So continue this analogy. If, for example, I crack this lens on my camera, I can just simply switch the lens and again continue to take nice pictures. In contrast, if I somehow drop the camera and the body of the camera is now broken and the processor doesn't work, well, it doesn't matter if I change the lens or not, it will still fundamentally take poor quality pictures. And it's very, very much the same at the level of the brain as well. If the problem is at the level of the eye, maybe a surgery like changing the lens, like in cataracts, or glasses, for example, can improve the quality of the image. But if the problem is at the level of the brain, the processor, then it really doesn't matter what you're doing at the level of the eye. It's just fundamentally not able to process the visual information correctly. So very, very important to distinguish these two aspects. Specifically about CVI, again, cerebral and also the other term you may hear is cortical visual impairment, has recently been given a functional definition, which says verifiable visual dysfunction, which cannot be attributed to disorders of the anterior visual pathways, i.e. the eye, or any potentially co-occurring ocular impairment. Now, that's not to say that in the case of CVI, problems at the level of the eye don't happen. They do. But on the net, when you look at the visual difficulties and the visual impairment that that individual has or that child has, it's largely driven by problems at the level of the brain as opposed to the eye itself. Visual acuity can range from normal to profound blindness. The entire range is possible, although most have some degree of visual function. Visual field deficits are often present, and typically that's in the inferior visual field. There is also evidence of reduced contrast. Ocular motor disorders may also be present. But the important thing to think about is that in terms of the definition of CVI, Impaired visual acuity or visual field deficits are not the sine qua non for this diagnosis. In other words, there are many children who have CVI who have perfectly normal visual acuity and no deficits in terms of visual field. As I said, it's really a fundamental issue in terms of processing information. Typically, in the case of CVI, there's a medical history consistent with pre or perinatal neurological complications. So in other words, something happened during development or during pregnancy or shortly after birth. And this could be some sort of hypoxic ischemic event, a stroke, for example, some sort of trauma or infection. And of course, there could be a genetic or metabolic disorders as well. And that's, of course, what ties into the conversation with CALD. Often there is a neurological comorbidity, cerebral palsy, epilepsy, for example, is quite common. And there are typically characteristic neuroradiological findings. So there are structural changes that we see at the level of the brain that give us evidence that there's actually damage going on. But again, similar to my previous comment, it's not a requirement for the diagnosis of CVI. So just because you don't see the damage on a structural imaging doesn't necessarily mean that the damage is not there or that the manifestation is not actually happening from a perceptual standpoint. And then here's the last part that I think is relevant to our conversation, is that often there is the presence of unique higher order visual processing deficits, what are referred to as visual spatial and complex motion processing deficits, as well as related to visual search and attention. Collectively, these have been referred to as dorsal stream dysfunction. So typically a child with CVI will tell you things like, sometimes I can't find a person I'm looking for in a crowd. Or if you give them their favorite toy, for example, they say, yeah, that's my favorite toy. They can identify it. But if I put it in a toy box, they can't find it. They have no idea. And often what these children will do is they'll take the toy box, they'll dump the toys out, we call them dumpers, and they have to space the toys out in order to find the toy that they're looking for. So clearly, this is much more complicated than just a question of visual acuity or a visual field issue. This is a processing issue, right? And we have to find a way to make sense of this and quantify it in a way that's actually objective and meaningful. So here is the fundamental problem. We don't typically test these aspects in a classic eye exam, in our ophthalmology exam, right? We use things like eye charts and perimetry or perhaps contrast tests. And even, for example, if we're doing things like cancellation tasks and things like that, these tasks don't really characterize the real world. They're very, very good for objectively measuring function, but they don't measure what's referred to as functional vision. That's to say how you use your vision in the real world. The problem with functional vision assessments is that we don't really kind of control things, right? The real world has lots of things going on. So if this child couldn't find their favorite toy, it could be for many reasons. It could be that many toys are there or present, similar looking toys, similar colors. We don't really know what the actual issue is. And what I propose is that there is a middle ground between functional vision and visual function, and that is the use of virtual reality, because virtual reality allows you to create scenarios where you have control of the environment, but at the same time, you can create environments that are what referred to as ecologically valid and behaviorally relevant. So you can create scenarios that are very, very close and simulate or an approximation to the actual real world. And there are two tasks that we've developed for this specific purpose. The first one is what's referred to as the virtual toy box. So this is a desktop task. You see here's the participant looking at a large screen at a toy box here, looking from like a top view. And what the child does is they pick their favorite toy. It could be a blue truck, a yellow duck, or an orange basketball. And this is important because we want to make sure that the child can identify the toy in isolation, right? And it creates an immersive feel for the task as well. It's then placed in a matrix of toys. And what we do is we change the number of unique distractors that are in the visual scene. And the thought is that as we change the number of unique distractors, the task becomes harder and harder to do, right? We can also do things like put another toy in the matrix that has the same color, but obviously not the same shape. Or we can add clutter to the background to see what happens when we make the visual scene very complex. But the point here is that we have now an objective measure that's behaviorally relevant for the child, and we use eye tracking as a way to measure their performance. So the child doesn't need to verbalize a response or use a manual response. We track their eyes to tell us how they're perceiving the visual world. And the reason why eye tracking and visual search is extremely relevant in this conversation is because we know that the network of the brain required to do these tasks is very, very extensive. It's not just a question of moving the eyes, it's deep structures in the brain that coordinate motion, visual cortex, parietal cortex, temporal cortex, frontal cortex, for example, for attention. So you can imagine that in the context of any type of damage to the brain, it will likely manifest at the level of this visual search and eye movement behavior. So that's really the connection between the two. So let me show you just some preliminary data of what this looks like when you use this task. So on the left, I have an individual with an age-matched control. On the right is an individual with CVI. And this yellow sort of blob that you see here is a recording of where their eyes are moving on this particular task. And the task here is to simply look for the blue truck on the screen. So let's look at that together. How about now? Well, if the computer was cooperating, what you would see is this yellow blob, for example, here moving onto the actual screen. And what you would notice is that in particular, well, the control would be able to find the blue truck very, very easily, but the CVI child, unfortunately, would be searching a lot more in order to get there. The important thing to take away from this is that this isn't actually related to some underlying eye movement issue. It's really more a question of the demands of the visual scene. So the fact that the visual scene is very, very complex leads to this impaired performance. And you can actually quantify this. So on this particular graph that I'm showing you here, on the y-axis, you have worsening performance. So this is visual search area, which is an index of precision of the eye movement. So the higher you are on this index, the worse you are. And on the right here, moving to the right, I should say, is increasing visual demand. So low, medium, and high. And what we noticed in our control participants when we do this task is that the relationship is largely flat, right? So from moving from low, medium to high, they do very, very well on this particular task. In contrast, our CVI participants do a little bit worse at the low condition, but as the task demands increase, their performance is actually much, much worse. And it's the steepness of the curve, the sensitivity to task demand, is what I think really characterizes CVI. And this is important clinically because when we're testing these children in a clinical setting, it's really under low task demands, right? But when you put them out into the real world, that's when the visual system seems to have the biggest challenges. So you need a way to kind of tap into that sensitivity of task demands and task load. The second thing I will mention is that even if a child has high visual acuity, so 20/20, 20/25, the performance is the same. So very good or near normal visual acuity does not get you out of this task sensitivity issue. It really is, again, reflecting the processing issue at the level of the brain. Again, I apologize for the videos not working there. Very briefly, the second task that we've developed is called the virtual reality task, and I will just again introduce this very, very quickly. Very, very similar to what I showed you at the level of the toy box. We have 3 task demands, except here, instead of looking for a toy, you're looking for a particular individual. This is the principal of a fictitious high school, and we increase the number of people in the crowd. And you again see the same sort of ideas that as more people are in the crowd, the visual search task is harder and harder to do. We can create other scenarios where there's other individuals who may have the same face but dressed differently. We can remove, for example, the clutter of the visual environment. But overall, the principle is the same. By tracking the eyes and seeing performance as a function of task demand, we can get a sense of how that individual is processing the visual information. And I'll hand it off to Melissa because she has some interesting data that she collected with the ALD population, specifically on this task that she'll share with you. And then just very briefly, I want to say thank you for this quick introduction. Again, a great collaboration that we're having with the Boston Leukodystrophy Clinic led by Dr. Eichler. And Melissa, I'll leave it with you now to share the data. So I'll stop sharing here. And I think you can go ahead with your presentation.
All right, so can everyone see mine? Yep, okay. So thank you, Dr. Merabet, for speaking to CVI and the virtual reality task. So now we're going to shift gears into talking about this in adrenoleukodystrophy. So just to introduce myself really quickly, my name is Melissa Bambery, and I am a clinical research coordinator with Dr. Eichler at MGH. And I currently am the lead coordinator on the ALD Bluebird trials. So I see a lot of Bluebird patients as well as other ALD patients in the leukodystrophy clinic. Okay. So quickly, I just wanted to say thank you to the study team. So Dr. Eichler, Dr. Merabet, and also Camille Kaur, who did a beautiful retrospective chart review that I will discuss before getting into the virtual reality data. So just kind of the main question that we're looking at for this is how is visual processing affected in ALD? So Dr. Merabet discussed visual processing as part of our presentation, but here's kind of a good visual to show you how visual processing might be affected in ALD. So here on the left is actually a brain MRI image of a patient with a posterior predominant brain lesion, a patient with ALD or cerebral ALD. And then over here, you can see that this lesion clearly falls in the area of the brain called the primary visual cortex, where this visual stimuli is being processed. So just to give you guys a little bit of background about cerebral ALD, both adults and children with ALD can go on to develop a brain lesion. When a lesion develops in childhood, this is known as CCALD or childhood cerebral ALD. When this develops in adulthood, this is known as ACLD or adult cerebral ALD. The presence of a lesion leads to brain demyelination, particularly in these occipital parietal regions, as kind of shown in the previous picture. Initially in boys, this presents with attention deficit or changes in visual processing. And then in adults, this can present with primarily cognitive symptoms in the early stages. So overall, these changes or deficits in visual spatial processing, it's referred to as cerebral visual impairment or CVI. So just to kind of go retrospectively, one of our coordinators, Camille, she did a retrospective chart review on these visual symptoms. So she took a look at all of clinic notes and neuropsychological evaluations from 2005 to July 2020. And she identified 89 patients with ALD and a lesion. So of these patients, 75% had developed a lesion in childhood, and about 25% had developed this lesion in adulthood. She looked at patients with varying less scores as well, so different size lesions, and of these 89 patients, about 60% had neuropsychological evaluations available for her to look at those symptoms. So after reviewing charts, we noted that there were a wide range of sensory processing issues or deficits. So here you can see if there were any visual processing deficits in the chart she noticed, there was about 50% had some sort of visual processing symptom noted. Specifically, we've noted that a lot of these were vision related, and most of these were very common. So the most common of these visual processing symptoms were the visual perceptual skills, field cuts, and visual motor integration. So that was clear that these visual processes were creating some symptoms. So overall, the takeaways from Camille's retrospective review were that symptoms were present in comparable rates in both children and adults. They weren't limited to a single pattern of lesion progression. When talking about an interior or posterior lesion, it didn't matter. They're prevalent even in patients with very early lesions. So like Camille noted, a less score of less than 3. And even after 2 years of transplant, symptoms could present as late as about 2 years following that transplant. So going into the prospective virtual reality study, Dr. Merabet already spoke to this, so I won't get too much into it, but just to reiterate, we looked at the 2 scenarios related to visual search and complexity. So first was to identify a particular toy among other toys and to identify an individual within a crowd. So in clinic, how I do this with patients is the first thing I do is calibrate the eye tracker for the participant. The next is I give them instructions to search for the target among the varying levels of difficulty or distractors, the number of the people in the hallway or the number of toys in the box. The target for the toy box experiment was the blue truck, and then the target for the hallway is the principal. And so the task for these participants is to look for and at the target whenever visible on the screen, while the responses are collected passively through the Tobii eye tracking system. Just to note, no verbal or a motor reaction was necessary for this. It was just collected passively. So for our study population, we had 28 patients with ALD do complete testing. Of these, 15 had developed a lesion in childhood, 7 had developed a lesion in adulthood, and 6 of these patients had ALD, but with no lesion on MRI. This was compared to 35 controls. So the virtual toy box task, Dr. Merabet spoke to as well, but you can see low to medium high difficulty. There were a varying level of more toys, 7 to 9 in the high density versus the low, 1 to 3. For the hallway task as well, the higher the difficulty, the more people in the hallway. So here's just a quick little video of what the hallway experiment looks like in real time. So you can see here, this blob is actually the participant's eye movements that the bar is picking up. So you can see right here, the participant finds the principal and identifies the target. So you can see the eyes are tracking the target all the way down the hallway until she passes you. So also just to show a high level of difficulty scenario, you can see here that there are at least 7 to 10 people coming down the hallway, and the participant here actually misses the target. They tried to find her, but she's over here. So that's just to give you an example. So just to start talking about a little bit of data, here the first data that's important for us is the mean off-screen counts. So this is the number of data points per trial that were off screen. So it's an index of task compliance. Here, the controls, there was about a 4% off-screen count and a 6% for the ALD group, meaning that there's only a 2% difference in between the two groups, which means that the ALD group in general, overall, the whole task, they were on task and they were able to complete the task. So going forward, the data that I'm going to show you, this means that it's not necessarily related to their ability to do the task. It's actually related to these visual processing symptoms. So here are 2 other points. So the success rate and the mean reaction time of ALD group compared to the control group. So the success rate is the percentage of trials when the participant successfully found and tracked the target. So found the principal and followed her with their eyes down the hallway. So ALD group, it was 94% success rate and control 100. So about a 6% difference. So a slight difference there with the success rate. Reaction time was the time from the start of the trial until the participant's eyes hit and remained fixated on the target for 400 milliseconds. So you can see there's also a slight difference here. This is very early preliminary data. So it's suggesting that the ALD group in general is less likely to find the target and a little bit slower when they did find the target. Then I wanted to talk about mean gaze error and mean search area. So mean gaze error is actually a measure of accuracy. So it's the difference between each individual eye data point and the corresponding target location. So you can see here, there's a difference between ALD and control groups. So the eye movements to find the target for the ALD group are a little bit less accurate, as well as a little bit less precise. You can see here. This is the mean search area, and so it's ellipse of best fit incorporating 95% of the data points. So then just to talk about the influence of having a brain lesion or not. So we had 22 patients who had a lesion and 6 we wrote as non-cerebral or NCALD. So these are patients who did not have a lesion. There was a clear difference between the control group and patients that had a lesion for this accuracy and precision measure. So this was statistically significant, how accurate and precise they were when compared to the control group. You could see here that the non-cerebral group was also similar to the having a lesion group, but we do have a low number of participants for the non-cerebral group. So I think this is very preliminary data, and hopefully when we have more participants, this will be more clear, the differences between the two groups. But right now, not significantly, statistically significant. And then I wanted to talk about the influence of task difficulty. So Dr. Merabet spoke to this too, but as the difficulty increases, so does the performance for these patients. So just to split up the patients with a lesion and without a lesion, you can see overall, the participants who had a lesion performed worse throughout the different difficulties of tasks when compared to the group without a lesion and the controls. So overall, this medical chart review revealed prevalence of visual symptoms. Testing of visual acuity alone does not capture these visual deficits due to having a lesion. Most of these patients we actually did testing with had 20/20 vision. So it shows that there is something going on with visual processing. Right now, there's no predictive tool or genotype phenotype correlation to anticipate which ALD patients will go on to develop cerebral AD. So it's a great way to measure these visual deficits, having this. And our prelim data propose functional visual assessments that can be used in the clinical setting to detect early visual processing deficits. So just a couple conclusions on this prospective VR testing. These were well tolerated by children and adults. It was about 15 minutes long, and we had both adults and children able to do this. We're able to do this in ALD. Obviously, the data is a little bit skewed towards patients with who are less advanced in their cerebral ALD, just based on the nature of the task. The dynamic hallway task seems to be a little bit more difficult overall than the toy box task, which could indicate a role of complex motor processing as an index for higher order visual spatial processing. And this task has the potential to pick up these higher order visual spatial processing deficits that might be missed through other assessments such as visual acuity, for example. So going forward, we would like to do more analysis of ALD patients that don't have brain lesions, and we're doing that now. So that NC ALD group. Longitudinal assessments we're looking at. So we have 4 patients right now with assessments more than 2 years apart. So maybe trying to track that data over time. It would be an interesting way to look at it. Comparisons with lesion load. So less scores in volumetrics and process. So comparing the performance with the less scores to see if there's a correlation there. Also, a comparison of ALD with ADHD patients might help differentiate CVI from attention deficits. And that is it. Thank you, guys.